Hi, everyone. Jerome, a quick logistical question. Is every meeting going to be under a new thread?
Every meeting under a new thread. So I was proposing that every time we start a new meeting planning, we create a thread in a discussion forum, and had the content, the agenda, and then anybody that has content to discuss, they just post it on the meeting thread. Does
Okay,
that answer your question?
it does, but it's becoming harder and harder to keep track of the many threads where people post
Yeah.
things. So, for example, I did not become aware that there was a new meeting thread for today's meeting until Nicholas, my student, alerted me that the agenda started to be posted under that thread. So,
Yes.
uh, I don't know when a new thread is initiated. When are we supposed to learn about it, and how we should learn about it?
Okay. Uh, I think I was hoping to follow this simple structure where at the end of the meeting today we'll discuss the agenda for next week, and then we'll create a discussion thread for next week. And then that's like the one place you might need to monitor for the next meeting. And
Okay.
there might be over threads if people are discussing analysis outside of meeting, but I think that's what I was proposing as a structure.
Yeah, I
Does
mean,
that make
I'm
sense?
happy, of course. Yeah, it makes sense. It's just that you know, I'm just trying to stay on top of things, but it's becoming harder and harder with everything else I have to do. So I'm relying on
Yes.
Nicholas really to keep track of things because I'm just not able to keep track of, there are at least three separate discussion threads where the analysis of the other data has been posted, and yeah,
Yeah.
maybe some others share the same thing, but I'm seeing people post on the chat. Yeah. Anyway, I'll
I
shut up.
I think it's gonna be hard to follow up everyone's work, but at the very least, I think the one that is the most valuable to track is the next meeting thread. I think.
Okay. Yeah, leave it to the community to decide how things should be done. Right.
Okay. I think we have forum. Um, Good morning, good afternoon, wherever you are. Today we want to continue the discussion we had last week. And so we have the agenda that is listed on this page. And Carter is going to start. So last week we discussed the confound of bleaching on SLAP2 data. And Carter is going to introduce a notebook and then the new data set will be uploaded to help address this confound. And then last week, Lucas did not have enough any time to discuss some of the analysis he did. Uh, we will give him the time to present his analysis so far, and we can have a discussion after that. I will present an update on bonsai development, is particularly something that I call the experimental launcher. And then we'll keep time for potential discussion of stimulus change that could be done on SLAP 2. And then we'll make sure to discuss next week meeting agenda. Are there any general questions before we dive into the presentation from Carter? Okay, Carter, are you there?
Yep.
Do you want to share your screen?
Yeah, let's see.
And this meeting is recorded, and we plan to post on YouTube. But so if you have any concern, please reach out before we do so.
Okay, can you guys see this?
Yes.
Okay. So on the website, we added this new notebook here. This, I think probably a lot of you have already, um, got the jump and started analyzing the new traces. But for those of us who haven't looked at it, I'm just going to take a little bit to explain more in depth what Casper was explaining last week, at least in terms of the files that we have. So we have new files on Dandy that have the F0, the DF, and the DFF, and they're named accordingly now. Okay, so this notebook should be able to just run out of the box. It's on the website, but I think it renders the markdown a little bit differently depending on where you're looking at it. So it might be better to look at it or to download it maybe, or to look at it on GitHub. So I'm going to look at it here. Okay, so this just sets up the environment, downloads the files. Here are the main ones we're interested in, right, that we have on Dandy. And it opens it. And the paths, so for DF, F0, and DFF, like Casper explained last week, it's basically the DF is the actual, preprocessed recording of the fluorescence that we see, right? And then F0 is essentially a metric of the baseline of the fluorescence throughout the session. So you can see bleaching, and then DFF is basically the trace accounted for that baseline. And I believe, I might need to verify this, but I believe they do have the same length in terms of timestamps, because they come from all the exact same DF recording. And they can be accessed like this. So you can pick which DMD you want and essentially just use these paths to grab the 3 different traces. Then for a given ROI here, I just picked one. You can view all 3 of these. So that's what they look like. Zoom out here. Yep. And then just in case you are wondering exactly if you want to play around with it or if it's relevant to your analysis, this is basically the code to reproduce how to actually obtain F0 and DFF. There are multiple ways to get F0, and the MATLAB code we use actually has several different algorithms. But I believe that the one we use for all of our data for this project is their first algorithm that they have. It's in a private repo right now, but I think if you reach out to Casper, you might be able to look at the code. So I translate, it's in MATLAB though. So if you're not familiar with MATLAB, this is translated into Python. So essentially it just does a couple of different smoothing operations. The main thing is a median filter, and then it's like a rolling window with a hole-like operation, a convex hole. And essentially what you get is this resulted super smooth trace. I might actually expect it to be more smooth than this, but this is what we have for the actual code that we used. And if you look at this trace and you compare it to the DFF in the file, it looks remarkably similar. Maybe except for the first and last values. And then once you have F0, it's very simple to obtain the DFF. It's just a matter of simple division, and you get the DFF trace here, which also looks remarkably like this. So that's it. If you have more questions about when to use which of these and how to use them, we can definitely continue talking about it. Casper knows more than I do, so you can ask either one of us. Yeah, that's it. Are there any questions? Okay, great. I think you're still muted, Drew.
Okay. Looks like you addressed the concerns. Thanks, Carter. So I think we recommend that all of the analysis are probably done in DFF if you want to make sure the fluorophore concentration decrease due to bleaching is taken into account. But I think we already knew that. Okay. Lucas, do you want to take it from there?
Yeah, sure. Yeah, let me share my slides. That would work. 1 second. Yeah, that's just a problem with permissions.
No worries.
Let's try. Okay, can you see my presentation?
Oh, we see a gigantic button. You're sharing your entire screen.
Okay.
What was the funny one?
um, okay, you see the presentation now?
Yes.
Yes, okay, perfect. Yeah, sorry, I'm not too familiar with Microsoft Teams. Okay, so I just wanted to give you a brief overview of an oddball analysis that I did based on code by Alex and Jerome and Sarah. And the idea here is that I essentially wanted to reproduce an analysis that was done previously, for example, by John Hamm in this PNS 2021, where he essentially compared pulse imaging of somas and found modulation of responses for deviant and redundant oddballs. So they would correspond to the deviant orientation or the standard 0-degree orientation in our case. And the question is, do we see something similar for the ROIs? So I wanted to essentially do these plots for the individual ROIs. That's shown here. So what I did, so this is, by the way, the latest data set here. And the what you see is here on the y-axis the mean z-score delta f over f. So I just included the new data that Carter shared and computed the mean z-score. The z-score is computed with respect to the mean and the standard deviation over oddball control 1 and control 2. So control, I'm referring the direction selectivity blocks, because they are the stimuli occur with random probability. So it's essentially, if you want the comparison between the individual, between oddball and control 1, oddball versus control 2, or control 1 versus control 2, that's all in delta f over f space. And the Z scoring is just a way to better compare between different ROIs. Okay, and what you see here on the right, so this is the dashed line indicates the stimulus onset, and then the second dashed line indicates the post-stimulus interval, but it's a bit arbitrary, but I just, or others as well, selected to essentially compare the responses in this window systematically between conditions. So what I did was to take the average over this post-stimulus interval for each trial respectively, and then one can do either average those again to compare means z-scores between the conditions, or to do some statistical testing, for example, comparing the means or the medians of these window averages. Yes, so essentially what I did is repeating this analysis for all the ROIs. Here's the DMD1 in this data set. And here what I plot is essentially the position of the ROI in the image plane. Then the size indicates the mean average set score in both conditions, essentially how strongly did an ROI respond to respective orientations, for example, 0 degree, 45 degree, 90 degree, averaged over all the control and the output block. And the color indicates the mean average set score difference between the conditions indicated here. So top row is the oddball minus first control or direction selectivity block. The second row is oddball versus the second control block. And then the last is the control versus control 2 versus minus control 1. Now focusing on the first two rows, so the oddball versus control comparison, we see that overall there are stronger responses for the 90 degrees. This could indicate that somehow inputs to this neuron are somewhat selective in this direction. But the contextual modulation is highly variable. So we see enhancement of deviant responses. So the 45 degree and 90s are the deviant directions. But we also see, for example, enhancement, so stronger responses for redundant inputs. And that kind of indicates that what we see here might be also due to noise. What I also did was to do statistical testing. Actually here, I say unpaired p-test. I also did a Mann-Whitney U-test on the medians. So a mean and a median comparison, and only plot ROIs here that are significant in both tests, and with a p-value smaller than 0.005. This is kind of an arbitrary value right now. One could see it as a multiple Bonferroni multiple comparison correction for a single ROI, more or less. But for now, it's just a way of filtering essentially noisy results, so we can focus a bit on what we think are proper effects here. And what we see is that there is more enhancement for the strongest deviation of the expectation. Deviation from the expectation should be the 90 degree orientation. This is already kind of consistent with what one might predict or expect. But overall, I was actually surprised to see so little depression, because this is the effect that you find robustly layer 2, 3, layer 5 for a lot of RIs if you do image somatas. So I was surprised to see so little depression for the ROIs representing synapses. And then the question here is also if there these what we see as connection modulation. So when we compare these blocks, because the oddball block is pretty long, if some of these effects could also be affected by slower mechanisms, for example, plasticity, so that responses systematically change over the course of time due to the oddball stimulation. And then when we just aggregate data as control 2 or control 1 oddball, we see some of these long-term changes also reflected here. And I just want to go through 3 examples, highlighting a bit the breadth of things that one can see and how we should interpret it. So I just wanted to highlight 2 cases of enhancement here, where we see like 2 of these red ROIs, where responses are enhanced, and we see this enhancement also reflected in a long-term fashion between the control 2, the later control block, and the first block. So let's look at those guys. This is one that shows a very nice deviant response, and you see that the control 2 response shown in darker gray here is also enhanced with respect to the first one. This is similar behavior here. One sees a bit of an enhancement here for the deviant direction compared to the control. And this is also reflected in the long-term change between control 1 and control 2. Now, there's also suppressed redundant response that also goes along with the long-term change. For example, here, so this is now one case of suppression. This is the response to the direction in the first direction selectivity block. And you see it's suppressed in the oddball. And this suppression we also see long-term if we compare control 2 versus control 1. The second response is much weaker than the first one. And then there's also a case of essentially no context modulation in the first comparison, but a long-term change if we compare the responses between the control blocks. So here we see no contextual modulation, but then throughout the oddball block, a certain response develops, and can be seen in the control 2. That's actually quite interesting that we have like an on and an off response depending on the orientation and so on. I mean, there are people looking into this in more detail. I just wanted to say there are these different effects of, let's say, short-term contextual modulation and long-term changes, and they somehow interfere, and we have to be careful interpreting these results. Just to wrap this up, I just quickly wanted to show you the epical at dendrite. I think it was ED2. And you see we have the same thing, kind of a high variability, but if we only plot the significant ones, if you want, then we again see very little depression, or in general, very little contextual modulation for this imaging plane. So to wrap this up, I found it surprising that there's so little depression for the standard stimulus. I was also looking into the other data set that we have. It shows in general less contextual modulation, but preliminarily one could conclude there's little contextual modulation in the apical dendrite. One saw indications of enhanced responses and long-term changes, and questions or hypotheses that one could come up with. Is there more potentiation for the standard direction, and what determines potentiation for enhanced deviance? It looked a bit like an enhancement in the oddball block also means an enhancement on longer timescales, but this is of course things we would have to test if there's more data. So next steps for this analysis would be to the statistical testing with proper multiple comparison correction. Also to link this to analysis of other people relating more systematically oddballs and direction selectivity, and disentangling the long-term changes. So plasticity from context modulation. And that's something I think we could maybe do through an experimental design. I wouldn't know how to do that best with an analysis, but it would definitely be interesting to systematically relate the contextual modulation we see in the oddball block and the long-term changes that we observe. Okay, I also have some notes here on the stimulus design, some thoughts, but maybe we separate that.
Okay. I have a question, Lucas.
Yes.
And I'm sure others have others. You mentioned that you didn't see much depression, right?
Mm-hmm.
And I wonder if this could be because the depression is very fast. So I recollect that some of the analysts in Jordan's paper in his lab, like when you present the drifting gratings, it just only take a few gratings for the depression to occur, and then it could stabilize really quickly. And so I wonder if that warrants a closer analysis where you're looking at the individual trials when the oddballs when oddball blocks start, just at the onset, and then after each oddballs to see if the adaptations is actually very quick. Have you looked in that or?
I only looked into these trial averages here.
Yeah.
So I was just wondering, would you see that in these trial averages then that you have? So what you were saying,
No.
fast depression even within
Well, let's say we have thousands of standard conditions, right? And if the depression is very, very quick, then boost will be washed out.
But wouldn't you then still see the difference between the oddball and the first control? If the adaptation is because of the repeated representation?
Yes.
Then
during the orientation block, you will see
earlier.
stronger. Yeah.
I mean, yeah. Because I mean, it could occur very quickly, but then I would still expect it to be less for the smaller response for the oddball. This is the first control. Or do you mean it's even within this window that it's already depressing? And that's why.
I think I was thinking every time the drifting is presented. I think you're making a good point, but the orientation block should have a stronger response.
Mm.
But I think we should take a look at the individual trial by trial presentation strength.
I mean, in a sense, I do that through the statistical testing. So that's really the averages of each trial. And then if there would be systematic differences between the trials, yeah, maybe we can discuss this again, but it's also comparing it to exactly the analysis that Jordan implemented. But I think just applying this analysis to his data, you would see more depression, I think. But I will check. That's a good point.
Yeah.
I have a question.
Yeah.
So I think I don't know how many samples of each stimulus was presented from which these were generated, but I would say that the order of the presentation of the samples, as far as the plasticity questions, is very important. For example, if you present a stimulus, let's say a 0 or a 45, and then you don't follow up on that same stimulus for many trials where other stimuli are being presented, cells, I mean, at least some of our published work showed that cells do care about the temporal time interval between presentations of the same stimulus, in the sense that the closer the stimuli are, the more sensitized the cells become, whether depressed or enhanced. So this is something important. So the trial order and the interval between the trials is important.
Yes, definitely. Yeah. I mean, one can also see that if one does the comparison here between the controls, there's, I mean, it's hard to say, but it seems a bit more potentiation actually in the standard direction that has more of this, stronger stimulation. It would be interesting to see if we see enhancement in the deviant directions, if that is really dependent on if we have deviant responses here, because if there's a strong salient signal, that could also be something that has a high influence on or strong influence on the plasticity potentially, right?
Yep.
But I agree. Yeah. Maybe also I can talk to Nicholas also about his analysis and how one can maybe get a better way of relating the oddball responses and the plasticity.
So these are, this activity, of course, is just a readout of a bunch of neurons, the activity of a bunch of presynaptic neurons, right? Neurons that are presynaptic to this one. And you mentioned that this is at odds with prior work on layer 2, 3, and layer 5 cells. How can we follow up on that? How can we square that away? I am fairly certain that most of the neurons presynaptic to this neuron are layer 2, 3 cells from somewhere.
Even on the proximal basal, you would say, or
even where? Sorry.
On the proximal basal compartments.
On the proximal basal compartment. On the synapses that are within layer 2, 3. And even to some degree, and like on the processes that are within layer 2, 3, I am pretty sure that most of the presynaptic neurons are from layer 2, 3. If there are processes that dip down into a deeper layer, then the fraction of thalamic inputs, for example, goes up. And also, in the apicals, there's more of a variety. But perhaps you all know more about the anatomy than I do. But maybe Mada can speak to the actual exact depth of these. Maybe these are a lot of processes. The apicals are certainly in layer 1, but I would still guess that at least many, if not literally most, probably like the largest single fraction would be layer 2, 3 neurons.
Mm-hmm. Yeah. Yeah, I mean, it's a great point. In the end, it should be from. Sorry, there was someone saying.
The question is whether all of the ROIs are spines, and that's not. There's nothing morphological about the way that the synapses are. The putative synapses are segmented. They're segmented based purely on activity, but we know that most of them are in fact spines, even if they don't stick out to the side, and like even if they're not clearly imaged. In the 2-photon plane, like if they're sticking directly above the dendrite, they're probably still spines. They're just not well resolved. So yes, Jesse, you should think of them as the majority of them are spines. There's also shaft synapses, but most of them are spines.
I wonder if it might be worth reaching out to Jordan. Because he was involved in, and I'm sure he will be happy to sort of share his wisdom on this analysis. Because that's why it's related to my point on the speed of decreased response strength. I think in Jordan's design they changed the orientation really quickly because it adapted very quickly. That's what I recollect, and so he probably is going to make some comments along those lines, I would suspect, and that might influence your analysis a great deal. And so I think it would be wise to reach out to Jordan.
Yeah, that's certainly a good suggestion. Yeah, we should do that.
Because I think Casper is correct, but we are generally looking at the same cell that he was. Yeah.
Yeah. That's true. That's why I'm also so surprised. Yeah, let's. Shall I reach out to him, or you want to reach out?
Yeah, I'm happy to. I'm happy to sort of help facilitate, but I'm sure he will reply. Yeah.
Okay, great. Yeah, let's do that.
Any other questions for Lucas? In terms of stimulus, let's talk a little bit about the stimulus change potential here. I I wonder if I'm also thinking about some of the analysis that Nicholas has pushed on the discussion forum, and which shows clusters of spines that have concerted changes, and so I wonder if this warrants an experiment where we just present orientation blocks without the oddball in the middle to compare with. Does that make sense?
Mm. You mean to compare general adaptation
Well,
throughout?
where we could have like orientation block without, I mean, we could have two orientation blocks that are separated by a constant time, and then no oddball in the middle, and then another pair with the oddball in the middle to compare. Because I feel like you're concerned about I mean, there's a lot of other caveats with like bleachings and other effects that could influence the response. And so if the oddball is really causing the change, it should show when you don't present it. Do you see what I mean?
Yeah, it's a good point.
I agree with that suggestion because the time of the oddball block should be accounted for between the pre and the post in that control condition. So whatever was generated in the pre and remained, around as far as the cells return to baseline firing or whatever, noting the short-term memory that was generated in those cells after that presentation, without the oddball, following right away, that could be a good control. Yeah.
Okay. I think Alice is asking if, yeah, so we did two experiments so far with calcium indicator. And last week, yes. And so we'll have to package it. It's just a matter of effort. But we have done one experiment which I think is worthy to share with the somatic calcium activity. Lucas, do you have other thoughts on stimulus change that you think we should discuss?
Yes. So this this was just a draft of to summarize these ideas, but I had a couple of things I noticed during the analysis. So one was that we have just like this one visual prediction context. We have just one standard direction. There are other experiments where, as you already mentioned, the standard direction is swapped throughout the experiment. And that does allow to analyze the mismatch under different visual prediction context. And it might give us, yeah, I mean, it's a lousy control in the sense that you were mentioning, but maybe if one would have control oddball in one direction, control oddball in the other direction, and another control, one might have a way to differentiate between the different standards in the conditions and how that affected plasticity to kind of compare these things. So if I'm saying this because then one really has two conditions that differed in the way the neuron was stimulated, and just trying to link that to that point. But in terms of the oddball analysis, the cool thing is that one can compare standard and deviant versus control for the same stimulus unit. So you might have noticed in these plots, you had either the blue trace or the red trace because these orientations occur just in one context. If one would exchange them, then one could have orientations where one has a deviant condition, a standard condition, and the control condition, and can then really relate how depression and enhancement potentially relate to each other. I think the changing the predictive context might also be interesting comparison of a predictive context for the omission response, because currently we do not have a good way to compare that omission to anything else, because we do not have a proper control for this as far as I understand. So comparing between different predictive contexts would be interesting to see if the omission response actually depends on what was expected, or if it's just a general alert signal, so to speak. That would be interesting to know for these responses. And then small things. I think we should enforce a minimum amount of repetitions after which a deviant can occur. It doesn't have to be 1/8 probability to get these numbers here, but essentially enforcing at least 3 repetitions or something like this. Because I noticed a lot of instances where a deviant was followed by a deviant, or even 3 deviants in a row, and for example, the oddball component starts with the deviant orientation. These things, since we only have 40 trials per oddball, it might be a good way to really ensure that they had a proper predictive context. As far as I understand, we also need a control for the static, because it's hard to compare in the sense of comparing the response in an unpredicted context or neutral context. One could think about reducing the number of standards. It would probably be necessary to fit the change of orientation into the same. The flip of the standards into the same experiment. And it could also make it easier to differentiate between oddball, like pure context modulation, and later changes through plasticity. Right now, the oddball is so long that these plastic changes occur already during the oddball block, most likely. And then we have a bit of a confound here of long term in this pure context modulation. Just some things I noticed, but of course it's always a trade-off, and some of these ideas definitely also impair other things that we want to do. That's it.
I think these are a lot of good suggestions. I have 2 thoughts. I think we do need to make a change. For the next round, I probably would put a diagram with all the different stimulus blocks to repeat, and then all the design parameters, and where we can discuss deeply about it with the diagram, so that we make an agreed with numbers. One analysis that will be helpful in my mind is how many trials we need to measure the responses, because I don't think we have any at this point on SLAP2 data. Because I think there's gonna be inherent response variability. Some of it is just noise. Some of it is due to stochasticity of the imaging of spines, and so I wonder if would it be possible to do some analysis of how many trials, like 1 to 10, we need to make a measurement. And that would drive.
That's a good point. How many trials does one need? We could subsample what we have right now and see how some statistics change as we increase the number of trials. Ideally, you would want to reach a number of trial where things do not change anymore drastically. So we are essentially in a regime where the exact choice of trials does not matter anymore. Then you go as small as possible. But
Yes.
we would have to agree on a set of observables that we think are important, that we should essentially should be invariant to this choice of trial.
Well, I think subsampling is probably the right way. And you do the same z-score analysis you did and see if your result is impacted by different degrees of subsampling.
Yeah, that's a good idea. The question is only because it will be affected, but how do we quantify that in a good way? Maybe essentially proportion uh, of significant responses, either enhanced or suppressed or something. But my intuition or my feeling is that we would not see a convergence within the 40 trials. But I will definitely do this, and we can see.
Because yeah, okay. I think that would drive the change we'll make in the next round because because we came into this with that little sense of statistical power, some of it, like for instance from prior studies, but now that we have actual data, that would be important to drive the next change.
Totally, yeah.
Okay, so I propose we start a thread on the next stimulus design. I put together a little PowerPoint or slide with like the potential change that you made with like numbers, and then and everybody should wait on there with the numbers that we want to reach. And I think on that thread we should add some statistical power analysis that would drive the decision. If you could do that, Lucas, that would be wonderful.
Yeah.
And others are welcome to join in. Okay. Are there any other comments on this wonderful analysis that Lucas presented? Okay. In the next, I want to present a little bit of progress with bonsai work for neuropixels and mesoscope rigs. So because this is also a goal, and I spend a fair amount of time setting up a framework that I wanted to show today, because we have to deploy this experiment onto many more rigs. So what I was planning to do was to show you on a Windows computer how I envision it will work. So I need to log in. Okay. So the challenge there is that our previous infrastructure was relying on Python. So we were using a set of Python libraries, and I made the concerted change to move to Bonsai here because this is our long-term goal at the island. So there was a bit of infrastructure that I have to build to deploy Bonsai on many rigs. And so this is what I want to show you. So let me see if I can share this. Do you see my Windows machine? Okay, so we have a system at the island that run any Python script that I can deploy on demand. So I can push a path to a Python script to a lot of machines, and the machine execute the Python with a set of parameters that I provided as input. So it's like a command line system where you say Python call this script with its parameters, and then we execute this system. And that's it's something that runs on top of all of our rigs. We have hundreds of them, and so I needed a way to easily deploy a Python script and a set of parameter files that control the behavior. The second issue is that Bonsai is great to execute the experiment, but we have a lot of parameters that are attached to each experiment that needs to be tracked. So the number of repeats of all balls, or the number of baseline conditions, the number of receptor fields. So these are parameters that are really important for the session. And I wanted a system that would track these along with the data. And so I thought a little bit about this problem. And so Bonsai has the ability to read files that would influence the parameters, but it also has input key values that are provided. And so if you launch Bonsai from the command line, you can provide any parameters you want. And so what I ended up doing was something called a launcher. And so this is a script that it's a single file that essentially communicate with some of the ecosystem at the island. So it fetches rig parameters that are different for every individual rigs at the start of the script. And then it looks locally for the presence of Bonsai. And so if Bonsai is not present in the system, it's going to download it automatically and deploy it on the machine. And then it's going to use JSON files that is, I have an example here. So that JSON file is a sample, but it takes as input the repository where the repo to clone, the branch you want to deploy. So this could be a commit hash. So this way we have provenance, exact provenance of which version we run. The local path on the machine where you want to deploy Bonsai, the actual path on the repo to the exact script that we want to run. So in this particular case, that will be a standard all-ball workflow. And then the path to the executable, the script, and then just a list of parameters that you want to provide to Bonsai. And so this little JSON is something we can track with every experiment, and it allows us to monitor what we executed. I wanted to do a quick demo so that you understand what I mean. So on this machine, I don't have the folder. And so if I just launch this little test script, so all of this code is already on the repo that you're using. And so if you launch this test script here, it's just using the launcher. It's a very thin wrapper. So it takes this JSON and then send it to the launcher and execute what the rig will be executing. And so what you see here when you launch this is that at first it looks locally, okay, I need to execute this thing. Bonsai is not present, so I'm going to download it. So it downloads Bonsai, and then it sees that, oh, I need all this package. So it downloads all of the package that I needed. And then it does this, so that takes a couple minutes. So it set up the executable. But then it's going to go to the repository where the script is located, but it's in the JSON file. And it's going to download the script. It does this by cloning the repo on the first step. So essentially that automates the whole process of deploying the script on the machine. And so you could run this outside of the island and it will mostly function. And so I view this as a way to sort of easily replicate running scripts across machines. And so right now it's downloading Bonsai packages, so the ones that are specified for this script. And at the end of the Python script is actually going to execute the Bonsai workflow and start the experiment. So if Bonsai is already present on the machine, it just bypassed that step and go directly to executing the Bonsai workflow. So that's the first thing. And then at the end of, so it executes the experiments, and once it's finished, in that Python script, I have a little set of functions that are doing packaging. So where is it? The installation kit, yeah, so you see now it's actually starting the script. So it did everything. So it was like a empty machines, and so we're just downloading all the requirements and then start the script on the machines. And when this is finished, so I just stop it. Once this is finished, there's a set of functions that are save the outputs. And so what this does is it takes the output of Bonsai and create a package that we organize in the way we want. And then this will be what we will package in the NWB for you guys. So, and that's nice because it also allows to add Python functions that reorganize the data in the way we want as we mature the packaging pipeline. So this is what this is a little bit of infrastructure work that I was doing to deploy this on many machines. Are there questions on this? Okay, it doesn't like, I mean, it's the inner kitchen. The second thing I'm currently doing is developing a number of C# plugins to just talk with the Institute hardware, and so that's going to take a couple more days, and then I'll write Python script like these for the different audibles types, and then we'll deploy it on multiple machine. Okay. Any questions before we discuss the agenda? Okay, next week, I feel it will be helpful if we discuss the stimulus design, the next iteration. And I saw that Nicholas has made a lot of really interesting progress, and so I wonder if it would be worth that we discuss his analysis of PCA clusters. I wonder if other people have interesting things they want to show.
Yeah, I can present. Oh, sorry, I hear myself. I can present next week my analysis. There's still a few revisions I need to do
Yes.
to my PCA analysis. I need to revise a few figures, but I can certainly present next week.
Okay, that'll be wonderful. And any other folks that want to present any analysis they've done?
Sarah mentioned that she wants to present data as well, focusing on ROIs at the single ROI level.
Okay. So we'll put this in the agenda. Sarah had a question on the pupil. So eye tracking data was collected on SLAP2. We haven't analyzed it yet. This is something that we'll do later on. But it will be a separate. It's an AVI files that eventually will be pushed on CodeOcean, but we haven't done that.
Okay, Jerome, is there a chance that we could take a look at how the quality of the calcium imaging, the spiking of the cells, even though our camp is poor, but maybe we can do something about it. I think it's super important to just trying to tie in what the cell is doing given all these inputs. So
Yes.
even if it's not the best, but taking a look at that data might be useful.
I will work with Maddie and Carter to see about packaging this data. Okay, so next week, Nicholas will present, and then Sarah, and then we'll have a discussion on stimulus design. with maybe input from Jordan's lab and some discussion of statistics probably. Does that make sense? Does anybody else has
It makes sense.
inputs or questions before we depart? Well, thanks very much for everyone that presented. It was wonderful.
Have a good day. Bye.